guys, so my topic is gel electrophoresis and its uses. First of all, so far throughout the year we've learned how complex DNA are, and today, scientists have a better understanding on DNA and its genes. But before that, scientists would ask questions like, when is a gene expressed? Or, where in the genome could this gene be found? So they came up with this indirect approach called restriction fragment analysis. This would help to identify different nucleotide sequences of DNA molecules, and with this approach came different techniques, such as gel electrophoresis. Gel electrophoresis is a technique where it uses gel to separate nucleic acids or proteins to determine its size, physical appearance, and electrical charge. To your right, you'll see a picture of gel electrophoresis, and you'll see a clear box, and inside is the gel, and attached to the box are wires which gives off the electrical current, which will cause the DNA molecules to move down the gel, which will help determine the size, charge, and physical properties. To see a better picture of this, I drew up a diagram. So to your left, you'll see these purple test tubes, and the scientists will pour a sample of DNA into each well in the gel electrophoresis, and due to DNA having a phosphate group, they are negatively charged. So the DNA molecules will move down the gel towards a positive charge. So the DNA molecules will make its way down the gel. And as you can see, I color-coded the DNA molecules. The red molecules were able to make their way down all the way to the bottom of the gel, which signifies how fast these molecules are. And this determines that it has a high charge, it's spherical shaped, and it's small. Compared to the blue DNA molecules, it barely made it out of the well, and it's still in the beginning, and it barely touched the middle, meaning how slow these molecules are, which determines that it has a low charge, is rod shaped, and it's long. So an overview is this of gel electrophoresis is that a sample of DNA is placed in each well. DNA will move towards the positive charge, and the result of this is that the fast molecules have a high charge, they're small and is spherical shaped, and the slow molecules have a low charge, it's long, and is rod shaped. To get these results, the scientists will have to pour a DNA binding dye onto the gel and stick it under an ultraviolet light, which will make the DNA molecules have a fluorescent pink color, and it will show them separated by electrophoresis. So, its uses. Gel electrophoresis allows scientists to separate strands of DNA, which will make it easier for them to identify and examine the components. So it would help them to identify carriers of a specific gene or disease, help them to look at DNA fingerprints, like identifying a murderer during a crime scene or determining whether someone is the real father or mother. And also, it could help them to have a better understanding on evolutionary relationships. And that's all.